Welcome to this explainer. When we look at complex cellular biology, it's so easy to get totally lost in the weeds with words like karyokinesis and diploid. Well, today we're stripping away all that noise to look at the elegant, foundational mechanics of the cell cycle and cell division. Let's dive into this biological masterclass together and really refresh our understanding of how this incredible process actually works.
Let's kick things off with a pretty massive question. How exactly does a single microscopic zygote turn into a fully grown human body made up of trillions of cells? Every sexually reproducing organism on Earth starts its life as just one tiny cell. Yet here you are, a complex being with a brain, a heart, and skin, all functioning together beautifully. The bridge between that single cell and the trillions inside you right now is the fascinating process of cell division. To give you a quick mental map of where we're headed today, we're going to look at why cell division is even necessary, how DNA and property division work, interphase, which is basically the preparation time, the actual M phase of cells, and finally, we'll wrap up by looking at haploid and diploid chromosomes. Let's get right into it. So, starting off with part one, why is cell division necessary in the first place? Well, there's three primary reasons for all this biological heavy lifting. First up is growth and development. From that single zygote to your current height, your body needed to constantly manufacture new cells. Second, healing damaged tissues. Have you ever gotten a paper cut on your hand and watched it completely disappear a week later? That's your existing cells dividing to replace the damaged ones. And third, sexual reproduction. To create specialized gametes like sperm and egg cells, the body relies on specialized forms of cell division. Moving into our second topic, let's talk about DNA and what I like to call property division. To really grasp the mechanics inside the cell, just think of a parent dividing up their wealth. Like a parent splitting an estate equally among their kids, a cell absolutely must perfectly duplicate its DNA before dividing. If a parent just chops a property right in half, neither child gets the full value, right? A cell, our parent cell, has a wealth of genetic information stored in its DNA. It can't just tear its DNA in half and give a piece to each daughter cell. If it did that, the new cells would be missing crucial instructions required to survive. To solve this literal property problem, the cell undergoes DNA replication. This is the crucial process where a cell synthesizes an exact duplicate copy of its genetic material. It also duplicates its cellular organelles, like the mitochondria. Only by creating perfect duplicates can the parent cell ensure that when it finally splits, both new daughter cells receive the complete, exact same set of biological wealth they need to thrive. All right, that brings us to part three, interphase, which is really just the preparation time. Think of a cell's life cycle kind of like a human's. We spend years growing and preparing before we're ready to have kids, right? Well, in humans, a typical cell takes about 24 hours to complete one full cycle of division. But here is the truly astonishing part. Out of those 24 hours, the cell spends 23 of them entirely in interphase. That means 95% of the cell's life is spent just preparing, growing, and duplicating its internal wealth, rather than actually dividing. Interphase is broken down into three logical steps. Step one is the G1 phase, or gap 1. This is where the cell undergoes active physical growth and is highly metabolically active. Step 2 is the S phase, which stands for synthesis. This is the big one. This is where that DNA replication we just talked about actually happens. And finally, step 3 is the G2 phase, or gap 2, where the cell makes its final preparations and synthesizes the exact proteins it needs for the upcoming split. And this brings us to a totally crucial heuristic. You can think of it as the golden rule of the S phase. DNA content equals double, chromosome number equals same. Let me explain the mechanics here. During the S phase, the cell creates a duplicate thread of DNA, so the overall amount or mass of DNA doubles. But because that newly copied thread remains physically attached to the original chromosome, we still count it as the exact same number of chromosomes. The amount doubles, but the count stays the same. This rule brilliantly illustrates the difference between those two gap phases that flank the S phase. In G1, we have our initial growth, the cell physically gets bigger, and most basic organelles duplicate. But in G2, the cell's doing its final targeted protein synthesis. It's also the specific time when powerhouses like mitochondria and chloroplasts duplicate. Everything is now fully prepped. Now, for part 4, let's look at the main event, the M phase of cells. Remember that 24-hour clock we were just talking about? Well, here is the number one. After 23 exhausting hours of preparation in interphase, the actual physical splitting of the cell, the M phase, only takes about 1 hour to execute. It's a remarkably fast and explosive process compared to all that set out.
During that single frantic hour, the M phase executes in two distinct steps. First up is karyokinesis. Karyon means nucleus, so this is the highly orchestrated division of the cell's nucleus, where those perfectly duplicated chromosomes are pulled apart. Once the brain of the cell is split in two, the second step kicks in, cytokinesis. Cyto means cell, so this is the physical pinching and division of the cell's cytoplasm, which finally gives us two distinct, independent daughter cells.
Finally, we arrive at Part 5, demystifying chromosomes, specifically the terms haploid and diploid. The comparison between these two is actually pretty straightforward. A haploid cell, which is denoted by the lowercase letter n, contains only one single copy of each chromosome. In humans, the only haploid cells we have are our gametes, our sperm and egg cells. On the other hand, a diploid cell, denoted by 2n, contains two copies of every single chromosome. Almost every single cell in your body right now, your skin, your liver, your bones, is a diploid cell.
This all perfectly maps out our golden rule mathematically. Think about it. In the G1 phase, we have a DNA amount of C and a chromosome number of N. Passing through the S phase, the DNA amount doubles to 2C because we copied the genetic material. But the chromosome number stays exactly at N. It does not change to 2N during synthesis, and it remains 2C and N all the way through the G2 phase, right up until the cell finally divides in the M phase.
We've definitely covered a massive amount of ground today, but I want to leave you with one final provocative question. What happens to cells, like our adult heart cells or the neurons in our brain, that permanently decide they are just done dividing? Well, they actually exit this 24-hour cycle entirely and enter what's known as the G0 phase, or the quiescent phase. They stay metabolically active, doing their vital jobs keeping you alive, but they lock their doors to division. It's a fascinating protective limit of human biology and honestly perhaps a perfect topic for our next explainer. Until next time, keep learning and keep wondering about the incredible biology happening inside you every single second.